Constitutive promoters are great when you just want something simple in your design that'll be on all the time and aren't worried about dynamics or toxicity of your proteins. Often, however, you will want some regulation, and for this you'll need to use more complicated promoters. When we discussed constitutive promoters, we said that they exist in two states, one in which sigma factor is bound and one in which it is not. In non-constitutive promoters, additional proteins called transcription factors will bind to the DNA, creating more potential states. One of the most commonly used promoters is the PBAD promoter. This promoter binds to a transcription factor called ARA-C in two different configurations that depend on whether or not ARA-C is bound to the small molecule arabinose. Only one of those states will bind sigma 70 and promote transcription. The result is that this promoter only turns on when arabinose is present. Thus, you can turn genes on and off by adding a small molecule to the cells. The PBAD promoter part came from the region of the E. coli chromosome upstream of the arabinose catabolic genes. Though often it is unclear where the boundaries of a part lie, through trial and error you often can identify a snippet of DNA sequence that retains the normal transcription behavior of the genomic promoter. You can then use that sequence as a promoter part, placing it upstream of other genes such as insulin shown here. These small molecule inducible promoters are particularly useful, but other promoters have been identified that respond to other things. It is also possible to use experiments like those done with the J23100 series to create new promoters that depend on various transcription factors in the cell. You can also engineer proteins that regulate a promoter part to respond to different things. Non-constitutive promoters are used ubiquitously in genetic devices. Simple on-off switches are used to keep toxic genes off until it's time to test them. For example, if you want to watch the biosynthesis of a terpene like borneol, it will be easiest to study that if you use an inducible promoter to control the genes. This allows you to grow up all your cells to a standard non-induced state and have a distinct start time for when your pathway began expressing. Also, non-constitutive promoters are required to make the genetic circuits that define the early years of synthetic biology. These circuits are usually composed exclusively of transcriptional activators, repressors, and the promoter parts they control. Biochemically, there is very little going on in these systems except transcriptional control. Before we move on, I want to remind you that we are dealing with a chemical system, and will often black box details when we talk about complicated things. In practice, these promoter initiation complexes are complicated shapes obeying the rules of quantum mechanics, and reducing this complexity to simple icons and numbers often neglects certain details. It is very easy to forget that the actual structure of transcription initiation complexes has many discernible microstates, and we necessarily describe them with far less refinement than physical-chemical theories would allow. As a result, it is often necessary to use more phenomenological models to describe the behavior of a promoter than physicochemical ones. Like with the sigma factors, transcription factors are really looking for a sequence. They are forming complexes with the DNA in which stabilizing interactions are satisfied. They recognize patterns of DNAs, just as our enzyme recognizes patterns of substrates. We can capture this pattern in terms of structure-activity relationships, and for DNA binding, hidden Markov models are the current best-in-class approach. We won't learn how the algorithms for that work here, but DNA binding patterns are more commonly illustrated visually as sequence logos, which capture the same information as the more quantitative model. These models can be populated by doing experiments such as CellX. Here, a pool of random DNA sequences is selected by binding to a mobilized transcription factor protein, and those sequences that are bound get selectively amplified by PCR. After multiple rounds of enrichment, the surviving DNAs are sequenced, and algorithms can extract the patterns from such data. We often use the terms transcriptional control and translational control. Let me rephrase transcriptional control in a little more granular form. Outlined here are all the various genetic elements that can affect the amount of an mRNA in direct ways. Some of these, such as transcription factors and sigma factors, affect transcription initiation. Others, such as riboswitches and antisense RNAs, affect the elongation and termination phases of transcription. Small RNAs, and in particular eukaryotic microRNAs, can also affect the post-transcriptional processing of the mRNA. These are examples of post-transcriptional control, but they affect the concentration of mRNA and thus from a modeling standpoint are most usefully considered in the context of transcriptional control. From a genetics perspective, we can categorize these control elements as being either cis or trans. A cis element is a separate gene that encodes a protein or RNA that is produced normally. The product of that gene binds to the promoter or mRNA, forms a complex, and changes its mRNA concentrations as a result. Cis elements are the things necessarily on the same DNA as the gene being regulated. Promoters are upstream of the transcribed sequences and define the start of that sequence. Terminators define the end of the mRNA, and riboswitches exist within the mRNA molecule. Transcription factors are usually proteins, and they directly bind to a DNA and either activate or repress transcription. They often form dimeric complexes and often bind to other small molecules or proteins. In prokaryotes, it is very common for the DNA to become looped within the complex. Often transcription factors form higher order oligomers that cause them to bring together two or more different regions of the DNA. It is often the formation of these loops that turn a gene on or off by locking the DNA in conformations that either bind sigma factors better or worse. Other times, the transcription factor will bind to the DNA partially overlapping the -10 or -35 box and thereby interfere with the normal interactions with sigma factor. In eukaryotes, things are quite different. There is a large protein complex called mediator that is also in the mix. I mention eukaryotes here only to make it clear that what I'm telling you today about transcription only applies to prokaryotes. We'll discuss transcription in eukaryotes in a different chapter. Here is the DNA sequence of the PBAD promoter that responds to arabinose. In blue, we have the -35 box, and overlapping this sequence, we have the CAP binding site. This configuration in which an activator binds just upstream of and partially overlapping the -35 box is typical. Additionally, we have Ara-C binding sites at various places, which is similarly typical. When they physically occlude sigma factor binding, they do so usually by overlapping the -10 box, but it is common to see repressor binding sites as far as 300 base pairs in either direction from the transcriptional start site. Ara-C represents one class of transcription factor in which there is one polypeptide that binds to DNA and it also binds to a small molecule. Another class of transcription factor splits the sensing and DNA binding portions of the transcription factor into two different proteins that interact through phosphorylation cascades. One component, the histone kinase, binds ATP and can transfer the gamma phosphate to the response regulator. The response regulator in turn binds to DNA in a way that depends on its phosphorylation state. Whether or not the histone kinase does this phosphorylation reaction or not depends on some stimulus, such as binding to a small molecular ion. E. coli encodes 24 of these systems natively, but other prokaryotes often have far more. For example, the soil bacterium Shewanella has 88 of these systems. In general, environmental bacteria have more need to respond to their external world than do pathogenic or commensal bacteria, and as a result have more transcription factors such as these. This figure shows the different two-component systems in E. coli and Salmonella. They are largely conserved across the enterobacteria. They respond to various specific ions such as phosphate, magnesium, and iron, but also more abstract things like envelope stress and osmotic upshifts. Many two-component systems respond to specific redox species such as quinone. In response to these stimuli, each two-component system regulates the promoters of many genes. A small number of regulated genes is illustrated in this slide, but the number of actual targets is far higher. In native promoters, it is very common to find many operators for response regulators that are not intuitively related to the original stimulus in an obvious way. For example, flagellar expression is regulated by cell density. Though genetically the regulatory machine of bacteria all look the same, the devil is in the details. E. coli and Salmonella have all the same orthologs of these transcription factors, but subtle differences in wiring can give rise to differences in gene expression. In this example, we consider the regulation of the PBGP and PMRD genes. In E. coli, the FOPQ two-component system regulates PMRD in response to magnesium ion, and the PMR-AB system regulates PBGP in response to iron. These two regulons do not interact. However, in Salmonella, the PMRD gene also regulates PMRA, and PMRA inhibits transcription of PMRD. This all boils down to subtle base pair differences in the promoters, but single mutations in the right place of a promoter can have profound effects on gene expression. In this case, the expression of PBGP in response to exogenous ions is quite different between the two organisms as a result of this slight change in genetic wiring. In terms of sequence homology, there are multiple classes of transcription factor. We've mentioned the ARA-C and two component classes, but there are a few others. Another way of distinguishing the transcription factors is whether they regulate just one set of genes or many genes. Ultimately, this is not necessarily a property of the molecule, but more of its role within the cell. Some transcription factors, like LAC-R and ARA-C, respond to very specific chemical stimuli, which in this case is lactose and arabinose, and in response they control a specific set of genes associated with degrading those chemicals. In those cases, the transcription factors tend to be included in the gene clusters associated with the processes they control. There are also a number of pleiotropic transcription factors like CRP, which responds to glucose and other preferred sugars, LRP, which responds to the hydrophobic amino acids like leucine, FUR that responds to iron, and so forth. In some cases, the stimuli is something specific, like iron, but there may be hundreds of genes that respond to the stimuli. In other cases, the stimuli is something more abstract, like envelope stress. Additional pleiotropic transcription factors, such as IHF, FIS, and HNS, don't respond to specific stimuli at all, but are important in changing the conformation of the DNA and thus play critical roles in regulating processes in the sense that they are necessary for proper function. There's a type of experiment called footprinting, in which you can map out where a given transcription factor binds to a DNA. And that is how we get this sort of information that allows us to draw these diagrams. Sometimes there really is just one repressor or activator over an otherwise normal promoter. However, many operons in bacteria have many operator sites and thus integrate many cellular signals together as a sort of logic gate. When you look at one of these complex promoters, it is tempting to think of them simply. Though operators often have sequence identity and are readily identifiable within a sequence, that does not imply that they are compositionally modular. If you just place a bunch of operators together in a sequence randomly and ask whether you get complex behavior out, usually the answer is no. There are precise geometries involving the organization of these operators and binding proteins that currently are not understood. Thus, promoter engineering right now can only be done using large combinatorial libraries and is highly unpredictable. However, entire promoter regions can often be used as modular parts, and they typically will retain their native transcriptional profile in a new genetic context.